Hello, everyone. Welcome to today's Revive webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi, and I'll be hosting today's session on charting new frontiers in AI for antibiotic design. Revive is GARDP's education and outreach program. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All of our webinars are recorded and freely available to watch after the session on our website, revive.gardp.org/webinars. You can also read our series of articles known as Antimicrobial Viewpoints. Here, experts discuss various topics within AMR. We also have a resources section, which includes the Antimicrobial Encyclopedia. This has definitions on various important terms that you'll hear about in today's webinar. Some of these also include videos where experts give a further explanation of the terms. As always, today's presentation will be followed by a Q&A session. You can submit your questions at any time during the webinar by clicking on the questions button as shown here. We will address the questions after the presentations and do our best to respond to as many as possible. If your question is for a specific speaker, please do indicate their name at the beginning of your question. And please note that we won't be considering the raised hands in the Q&A session. Today's speakers are Jonathan Stokes and Thorn. Our moderator today is Akhila Kosaraju. Akhila Kosaraju is the CEO and president of Phare, a social venture using AI and deep learning to develop novel classes of antibiotics in partnership with Jim Collins' lab at MIT. Akhila has spent her career building companies and driving innovation in infectious diseases and computational biology. She was previously the founder and CEO of Phare, a venture-backed company specializing in genomics and therapeutic development, and an executive with Seeger Technologies that successfully advanced antiviral drugs candidates to market. Akhila co-founded the Alliance to End Biological Threats and is a lecturer at Stanford's Center for Biosecurity and Pandemic Resilience. She received her medical degree from Columbia University and her bachelor's degree in human biology from Stanford University. Welcome, Akhila. I'm happy to hand over to you to introduce our first speaker.
Hi, Victor. Thank you so much for the kind introduction. It's my great pleasure to be here, and thank you, GARDP, for highlighting the incredible frontier work in artificial intelligence for antibiotic discovery and design. And it's also my great privilege to introduce John Stokes. Not only has he had an extraordinary career at McMaster and starting Stoke Bio, he also was instrumental in the founding of Phare Bio. So always such a pleasure to join the stage with you, John. John is an assistant professor of the Department of Biochemistry at McMaster University. He's co-founder and CSO of Stoke Bio. His research group focuses on the development and implementation of machine learning methods for drug discovery and design. They're currently working on discovering novel antibiotics to overcome drug-resistant bacteria and new molecules to treat an aggressive form of brain cancer called glioblastoma. He received his PhD in antimicrobial chemical biology from McMaster University and was advancing postdoctoral fellow at the Broad Institute of MIT and Harvard in the US. I'm very happy to welcome you, John. Great to see you as always, and I'll let you take it away.
Awesome. Hey, everybody. I'm John. I work at Mac. I'm excited to be here today, like super excited. I'm super jet lagged. I got off a plane from Spain yesterday. So if this thing catches on fire, we'll get through it. We're going to be okay. All right. So I want to talk about how we use AI to find new antibiotics. I don't know why I put this slide here. I think it's more for me than anybody else. It kind of justifies my existence, right? We've had antibiotics for the better part of 100 years. The majority of them were found between the 40s and 60s in the golden era through natural product mining. And then come the middle of the 1960s, we couldn't discover antibiotics using this approach very effectively anymore due to the dereplication problem, right? So in more recent decades, we've turned to alternative approaches, high throughput screening, medicinal chemistry, et cetera. And then, we find ourselves in this discovery void due to, I'll say the suboptimal performance, let's say, of conventional, more recent, if we want to call it that, high throughput screening approaches. So we haven't discovered, a fundamentally novel class of antibiotics since the late 1980s. And this, the discovery void in and of itself is not necessarily a problem, right? We have 20-ish classes of antibiotics, and they worked. The problem is, what accompanies our discovery void is growing antibiotic resistance, right? So right now, I'm sure we all know, we have 1.5 million people a year dying due to drug-resistant infections. That's roughly one person every 20 seconds, and this problem is projected to grow to 8-10 million people a year dying due to drug-resistant infections by 2050. The other thing that we need to consider, though, is like not just overall mortality due to drug-resistant infections. It's the fact that antibiotics uphold a lot of modern medical practice, right? So things like immunosuppressive cancer chemotherapy, any invasive surgery, preterm births, etc., are only routinely practiced safely due to our ability to control infection, right? So, if we're in this space where, resistance will evolve to existing antibiotics, and we all know that there is, not a very strong incentive to discover new antibiotics, how do we discover fundamentally, structurally, and functionally novel antibiotics more rapidly and less expensively, right? And I think that's where the application of ML has a lot of potential. So, to put this in perspective, right, AI allows us to explore vast chemical spaces for novel, useful potential antibiotics. In a high throughput screening setting in a laboratory, we can look at millions of molecules at the largest scale, millions. And typically we have a less than 1% in vitro success rate. However, if we have an appropriately trained AI model with, high quality, biologically relevant training data, we can look at, more than billions of molecules, and based on empirical success rates, we can see greater than 10% success rates if we're looking for structurally novel stuff relative to, compounds that we've seen in training. My lab is, we're really interested in Acinetobacter. I really like Acinetobacter. It's a hospital-acquired gram-negative pathogen, and why I'm super interested in it is because we need new drugs against it pretty bad, right? It's multi-drug resistant, causes a lot of really nasty infections, and mortality is really high, 30 to 60% last time I looked. And what's interesting about Acinetobacter is it likes to take up extracellular DNA and just kind of hold on to it, right? So you have these really big plasmids, and you can imagine if you're an Acinetobacter cell living in, a hospital setting, you're going to acquire lots of multi-drug or antibiotic resistance genes, right? And Acinetobacter baumannii is also something that I call a professional pathogen, right? It lives in hospital settings and like doorknobs and stuff. And the people who get Acinetobacter infections are folks who tend to be in hospitals for prolonged periods of time. They tend to have other illnesses, and it opens them up to like these really aggressive, bad Acinetobacter infections. So, we want to kill Acinetobacter, and we want to leverage an AI method to help us do it. So I'm going to talk about 3 little stories today, and each one kind of builds on the previous. So, let's set out with like a really simple use case, right? We want to discover new drugs that kill Acinetobacter. Okay. The first thing we need to do if we want to train an AI model to be able to predict novel molecules that kill Acinetobacter is we need to go into the lab and gather appropriate training data. And that's what we're looking at here. So this is 7,500 compounds screened against Acinetobacter baumannii, just looking at growth inhibition. All the molecules in blue are inactive, and we labeled it with a 0. All the molecules in red are active. We label it with a 1. So what we're showing here is something that we can call a binary classification task. What we've done is taken, our complex primary screening data and just labeled every molecule with a 1 or a 0. Is it antibacterial or not? And what we have now is roughly now 7,500 structure property pairs that can be used to train an AI model, right? And there are many, many ways to do this. But fundamentally, all we need to do is figure out a way to turn the structure of all those chemicals into a vector, which is just a big string of numbers. That vector then serves as an input to a feed-forward neural network that outputs a single number between 1 and 0. So what does this mean? What we're trying to do is train a model such that it can look at any structure of a chemical that we show it and convert that structure into a number between 1 and 0 that reflects the confidence of the model of whether that input molecule is antibacterial or not, right? And like I said, there's 100 ways to turn the structure of a chemical into a vector. What I'm showing here on the left is something called a graph neural network, which is, an architecture that we've had lots of success using for these types of prediction tasks. And if you want to geek out about graph neural networks, send me an email. We can talk about it later. But once we have our trained model, right, we can now run predictions where we can show the model brand new chemicals it has never seen before, i.e. things that are not in the training data set. And it will make predictions based on whether each input chemical is likely to be antibacterial or not. And that's what we're showing here on the right. So this is just a plot of prediction score across about 7,000 molecules that we showed this model. And what do we see? We see a very small number of molecules have a high prediction score above 0.2, and the overwhelming majority have low prediction scores, right? And this fits with our mental model of the world, right? We know that most molecules are not antibacterial. A very few of them are, and that's what we see reflected in the distribution of property prediction scores from our model. So now what we can do is take all the molecules that have a high prediction score, right? Everything above 0.2, identify those molecules that are structurally novel relative to any known antibiotic, and then we can identify a molecule like abscin here that does not share, structural features with known antibiotics. We can bring that molecule into the laboratory, test it, and see whether it works. And here is the structure of the molecule. It had an MIC of 2 micrograms per mil against Acinetobacter, and it was a little bit bactericidal, right? It killed about a log. I'm not going to go into the details of the story. It's published, and it was picked up by a lot of people. A lot of people thought this was cool. I thought it was pretty cool. Not super cool. It was cool enough. But why did I talk about this? I wanted to use this as an example to highlight the limitations of discriminative modeling, right? So this idea of having a trained model, showing it pictures of new molecules and it having to predict which molecules are antibacterial or not, is really, really, what's the word I'm looking for? Inefficient, right? Because by definition, the overwhelming majority of the molecules we show it are not going to be antibacterial, like 90+%. So it's wasting 90+% of the time looking at molecules that are unlikely to have the property of interest, which in our case is trying to kill Acinetobacter. And at the largest scales, right, at least with computational infrastructure we have, we can look at like 1 billion chemicals for something like that using a discriminative model. But drug-like chemical space is 10 to the power of I don't know how many molecules, right? Like bar fights break out over this. I've heard people say like 10 to the 20, 10 to the 60, 10 to the 100. But I don't know. It doesn't matter. Like, for reference, our planet has 10 to the 18 grains of sand on it, right? So if it's 10 to the 20, that's a 1 with 20 zeros behind it. It's like that might as well be infinity, right? We can't look at that many molecules. But the question becomes, are there better methods to more thoroughly search 10 to the 20 molecules as opposed to using a discriminator where that's just impossible to do, right? And that's where this whole idea of molecular generation comes into play. So, we're not the first people to think about molecular generation for new drugs, right? This is an old idea. The problem is a lot of models that have pre-existed build molecules atom by atom. And by building molecules atom by atom, you run into this issue of synthetic intractability, right? Like you can have models that output molecules that are really, really pretty to look at on a computer screen, that when you show them to a synthetic chemist, they laugh you out of their office because they can't make it. Or if they can make it, it's ridiculously expensive. So, we're interested in developing generative models that can search broad chemical spaces for novel antibiotics, with the caveat that they have to be cheap to synthesize and they have to be synthetically tractable, right? Synthetic tractability is important because I want to bring these molecules into the lab and test them. And cheap is important because I'm an early career researcher and I'm broke, right? So all of this was born out of necessity. So I worked with, our lab worked with a buddy of mine, James Zoe, out of Stanford, and we developed this algorithm called SynthMol. SynthMol is a molecular generation algorithm that we applied to design novel molecules against Acinetobacter. And this is a very high-level overview of how this works. So the first thing we do is we gather a training data set, right? Molecules that inhibit the growth of Acinetobacter, we've already seen this. We train a property predictor on that data, we've already seen this. Nothing new. When it gets interesting, though, is that now instead of going onto the internet, finding structures of molecules to now show this trained model, we turned this into a molecular generation problem as follows. So what we did is we went on to the internet, found 130 plus thousand multi-atomic molecular building blocks. Think about these as like Lego pieces. And we found a collection of 13 chemical reactions, right? We know the structure of every building block. And across these 13 reactions, we know the synthetic compatibility of every building block with every other building block. And that gave us access to a chemical space of 30 billion molecules, right? So we then trained a Monte Carlo tree search, which we'll get into in a couple slides, how to navigate this combinatorial space of building blocks and reactions to synthesize molecules, right? Design molecules, which when shown to the trained property predictor, get a really high prediction score, meaning that the model thinks they're antibacterial, right? And because all the molecules that are outputted are synthetically tractable and cheap, we can bring a bunch of them into the lab and test them, right? So here's our training data set. Instead of using, a smallish training data set of 7,500 compounds, we got a little bit more. This is 13,500 across 3 distinct chemical libraries. And what we're showing on the bottom half of this slide is a 2-dimensional representation of the chemical space that's explored amongst these 3 libraries. What we really care about is the distribution of all of the hit molecules across these 3 libraries. What you want to see when you run this type of analysis is that you have structurally diverse hits, meaning that your hits are scattered across the totality of chemical space that's represented in your training data set. That's really important because you want your model to be able to generalize very well to novel chemical spaces in the search for structurally novel bioactive molecules, right? And that's what we saw. Importantly, we didn't invent the 132,000 molecular fragments and 13 reactions. We worked with a good buddy of mine from Enamine, who compiled these fragments and reactions as part of their real space. And this is available. I encourage you to go check it out. Okay. What I'm going to show you now is our molecular design process, like sort of at a high level. So this is a Monte Carlo tree search in a nutshell. This is how it works. So again, our goal is to design molecules out of this combinatorial space of building blocks and reactions, such that when we show a molecule, a design molecule to the property predictor says, yes, this is a good molecule to synthesize because I think it's an antibiotic. Okay, here's how it works. Level 0 is what we call the root, and it's empty, so ignore it. In level 1, what we do is we take every building block, all 132,000 building blocks, and we show them all to the property predictor that was trained using our training data. We then rank order all of the building blocks based on their property prediction score. And we just take the building block with the highest property prediction score from our trained model, and we say, if this building block has the highest prediction score, we're going to use this as a starting point to build a molecule from. So this building block now brings down to level 2, and we ask a question. What are all other building blocks that are synthetically compatible with our original building block that we prioritize from level 1? We then take every synthetically compatible building block and do the exact same computation. What is the property prediction score out of all synthetically compatible building blocks? Which one is the highest? And we prioritize it, right? So in this case, level 2, our highest property score was this second building block. And we can see that our first building block from level 1 and this prioritized building block from level 2 are synthetically compatible via reaction 11, whatever that is. We can then take these 2 building blocks and reaction 11, in silico synthesize the resultant molecule, show it to our property predictor, and that property predictor will output a score. In this case, the score was 0.9, which is very high, right? Meaning that the property predictor said, hey, this approach built a good antibacterial molecule. That score is then back propagated through the model and increases the scores of the nodes that went into building this molecule, right? Conversely, if this was a very, very low prediction score, right, that low prediction score would be backpropagated, and the scores of the nodes that went into building that molecule would be decreased, right? So what you do is you run the same approach like 20,000, 30,000, 100,000 times. And what you're going to have over time are various nodes that repeatedly result in making good antibacterial molecules and various nodes that don't. They make bad antibacterial molecules. Since this Monte Carlo tree search is trying to identify optimum paths through this combinatorial space in order to maximize the property prediction score, what it's learning are how to select building blocks in order to make molecules that are likely to be antibacterial, right? That's how a Monte Carlo tree search works. So we ran this. I think we did 20,000 rollouts, synthesized a handful of molecules, and they worked in the lab, and that was good, right? And now we have SynthMol version 2.0, which works better, that we're finishing up the paper for, but I'm not going to talk about it today because I want to talk about something else. Here's the paper. You can go read it, or you can just email me and we can talk about it offline. All right. I want to talk about one other thing that I think is super cool. I'm a phenotype guy, right? Like I'm not smart enough to know what a good antibiotic target is. But I know like if I'm trying to kill a bacterium, I need to like that's the phenotype I'm chasing, right? The problem is, right, is if you have a structurally novel molecule that has antibacterial activity, you often don't know how it's working. And figuring out mechanism of action is like a bit of a job, right? It takes like a year and a half more from like a single grad student working all day, every day. So I want to talk about a project where we leveraged an AI model to help us predict mechanism of action, which I think is really, really important. Here we're not talking about Acinetobacter, we're talking about Enterobacteriaceae, specifically adherent invasive E. coli, which is a pathogen that's like commonly associated with inflammatory bowel disease. All right, so I haven't talked about this before, so this is like everybody's getting an early look. This was done by Denise in my lab. The paper's in revision. So we screened a whole bunch of molecules against adherent invasive E. coli, identified hits, and by imposing, growth inhibition thresholds, structural novelty thresholds, other molecular feature thresholds, and a literature search, we found this one molecule called enterololin, which was not discovered by an AI model. It fell out of like a really cool primary screen. What was really interesting is when we took enterololin and tested it. Hang on, I'm trying to get my slide to switch here. Sorry. When we tested enterololin against AIEC in the laboratory, right? Again, this is lab data. The MIC was 16, which is not great. When we tested it against BW25113, the MIC was 32, which is not great. However, we did an experiment where we used a hyperpermeable efflux deficient E. coli, and now the MIC dropped to 30 nanograms per mil. So we had a molecule that just had a permeability issue, right? And that was recapitulated here. We have AIEC in the presence of SPR741, which is an outer membrane perturbant, and we get our 30 nanogram per mil MIC back. So you're like, okay, this is rad. We have this molecule that has an inherent outer membrane permeability issue, right? Join the club. There's a ton of them, but we know when it can get to target, it's really potent. So we started studying this molecule a little bit more. And it turns out that it's Enterobacteriaceae specific, right? It has an MIC of 16 against Kleb, which is bad, but it doesn't have an MIC against Acinetobacter pseudo or staph. And why we care about this in the context of AIEC infections is because you can't treat an AIEC infection with a broad spectrum antibiotic, because broad spectrum antibiotics, as we all know, induce dysbiosis. And what happens if you have an IBD patient who has an AIEC infection and you treat them with a broad spectrum antibiotic? That dysbiosis promotes chronic and recurrent AIEC infection. So this is one of these diseases that necessitates a narrow spectrum antibiotic. And it appears that we, potentially have an Enterobacteriaceae specific compound. Indeed, this is largely recapitulated when we perform in vitro checkerboard assays where we combine enterololin with SPR741, and we observe the most potent synergies in AIEC and Klebsiella, which are both Enterobacteriaceae. We have a tickle of synergy in Acinetobacter and nothing in Pseudomonas. When we take a collection of clinical isolates, this also proved to be true. We took a collection of multi-drug resistant isolates of, broad phenotype or broad phylogenetic diversity, and this held true. So this is where the project got interesting. The students who are working on this, they're way smarter than me. And they said, well, we have this really interesting phenotype, which is Enterobacteriaceae specificity. Are there any other molecules that exist in the world, right, that have Enterobacteriaceae specificity and share structural features, however minor they might be, with our parent molecule enterololin? I can't share you the structure of enterololin because the lawyers at McMaster won't let me yet, but we could talk about that offline. I apologize. But it turns out, if you look at the structure of enterololin and you compare it to all the other molecules that exist that are Enterobacteriaceae specific, they share this sort of dual ring structure here in blue and green. What I'm showing here, this TSE, this just means Tanimoto similarity to enterololin. And Tanimoto goes from 0 to 1. 1 means it's the same molecule. 0 means it's super different. Anything below 0.5 is considered like a fairly novel molecule. And if we look at the structural similarity of all these known molecules that are Enterobacteriaceae specific that also share these structural features with enterololin, the Tanimoto similarity is super low, right? So they're structurally completely diverse. But what's really cool is all of these 5 molecules share a target. They all inhibit the LOL-CDE complex in Enterobacteriaceae. The LOL-CDE complex is involved in transporting lipoproteins from the inner membrane to the outer membrane. So we had a hypothesis now. We have phenotypic similarity, i.e. Enterobacteriaceae specificity, these really interesting structural features that are common. So does enterololin potentially target the LOL-CDE system? And to do that, we collaborated with a really good friend of mine, like maybe my favorite person, Regina Barzilai from MIT. She's just the best. She's a computer scientist who had recently, around the same time we discovered enterololin, published a model called DiffDoc. DiffDoc is a diffusion model that can predict the binding of small molecules to proteins. So we called up Regina and said, hey, we have a molecule. We have kind of an iffy hypothesis that this compound could be binding to the LOL-CDE complex. Can we test this? And we did it. And it took 100 seconds. And it put enterololin in the business part of the LOL-CDE complex, right up here. So we said, okay, this model, which is an ML model, predicted that our molecule sticks to a really important part of this protein complex. Do wet lab experiments validate this, right? So we went into the lab, evolved a whole bunch of resistant mutants, and they all mapped to the LOL-CDE complex directly beside where DiffDoc put the molecule, right? So we were like, okay, this is super cool. Like all of our resistant mutant mapping agree with the DiffDoc predictions. Can we now use orthologous methods to add additional validity to this hypothesis? So we go into the lab, do a whole bunch of more experiments, which took 6 months, right, as opposed to 100 seconds. So we went into the lab for 6 months, did, fairly standard, bacterial viability assays. We then did an RNA-seq experiment, asked do the transcriptional signatures of wild type AIEC treated with enterololin, are they consistent with inhibition of lipoprotein trafficking inhibition? And the answer to that was yes. And then we did a simple CRISPR experiment where we knocked down expression of LOL-C, which is a protein in which we were getting most of our mutants. And we asked, if we knock down LOL-C, do we see enhanced potency of enterololin? And the answer to that was yes. Right? And this is consistent with other known LOL-CDE inhibitors. Right? So this was a case where we got to leverage an ML model to help us predict the biological function of a novel molecule based on like a couple subtle clues, right? And then, of course, we had this molecule. We showed that it's non-toxic. It can kill intracellular AIEC, which is important. This is just AIEC being killed inside macrophages where AIEC likes to live. And then we can take the molecule, put it in a mouse, and then when we combine it with SPR741, we get significant significant clearance of the AIEC infection in the guts of mice. It's not perfect, but it's a really, really good starting point, and we're continuing to optimize enterololin based on these results. So, oh, my screen just went black. I apologize. Anyway, that's all I had to say. So my next slide was going to be my thank yous. Everybody, you know, I answer emails and apply for grant funding all day. The students in my lab do all the work. So I just want to give a huge shout out to them. And, you know, you can check them out on my website. We also released this free tool. If you go to escape.mcmaster.ca, a really talented student in my lab, Autumn Arnold, made this website where you can go and run your own predictions on any molecule you put in across all the escape pathogens. So give it a test. If something breaks, email us, and we're always trying to fix this and make it better. And then with that, I think my next slide is actually my thank yous. Yep, here it is. So here's the group. They're all way smarter and better than I am. And thanks to everybody who enabled this work through funding. And here's all the funders. All right, that's it for me. Thank you so much.
Fantastic presentation, John, as always. And I see some really great questions in the chat here. We'll push all of them to the end, to the Q&A section, but I'll make sure that we direct these early questions toward John. So it is also my great pleasure to introduce Kurt Thorn. Kurt is the Chief Technology Officer at ArrePath, a biotechnology company using a combination of human intelligence and cutting-edge AI to discover and develop small molecule therapeutics to address significant unmet medical needs. He leads efforts to develop tools to accelerate drug discovery for novel antibacterials. Prior to this, Kurt was Senior Director of Data Science at Zymergen. Before transitioning to industry, he was a research fellow at Harvard University and an associate professor at University of California, San Francisco, and director of the Nikon Imaging Center at UCSF. He has a PhD in biophysics from UCSF and received his bachelor's degree in chemistry from Princeton. Welcome, Kurt. We're really looking forward to your presentation.
Great. Thanks so much, Akhila, and thanks, John, for that great talk. So I'm going to talk about our work at ArrePath using ML to identify antibiotic candidates that we can take forward to develop into drugs. And in many ways, this is going to be very similar to what John talked about. We have a slightly different spin on how we do things, but the overall goals are very, very similar. So I don't need really to give much introduction here since John's covered the background quite nicely, and I'm sure most of the folks in this webinar are familiar with it. But you know, we've not discovered new antibiotics that have made it into the clinic in quite some time. And a big barrier here has been the permeability challenges in gram-negative bacteria. So like John showed with his enterololin molecule, it's much easier to identify molecules that kill bacteria when you defeat the permeability barriers in the efflux pumps in those bacteria. But when those barriers are intact, getting molecules into the cell where they can act on their targets and kill the bacterium is quite challenging. And so ArrePath was founded to address this problem using a three-pronged approach. And so one thing we've been doing is using machine learning models to predict novel compounds that are going to be active against intact bacteria. We do phenotypic screening using a combination of high throughput growth assays and then imaging to then identify or predict the mechanism of action of those compounds. And in this talk, I'll talk mostly about the ML modeling approaches we've taken, but I'll briefly mention the imaging approaches as well. So here's a traditional approach on the left that you might do. You run, you know, some chemical feature selection to select molecules in a drug-like chemical space. You run a large-scale growth, no growth HTS screen, and then you figure out what those compounds do through some kind of chemical genetics approach, similarity mapping like John just showed, other things. On the right side is what we've been doing at ArrePath over the last couple of years. We do ML up front to predict which molecules we should screen in the first place. And I'll talk a lot about how we build those models and use them. We then run those high throughput screens to find those compounds that inhibit the growth of the bacteria of interest, take the hits, run them through our imaging platform to look for ones that are phenotypically novel, and then go on to profile those compounds in more detail, find targets, and advance the relevant ones into hit to lead and lead optimization. And we're doing this all in gram-negative bacteria, and in particular, Enterobacterales. And so that the key hypothesis underlying this work is that ML models can learn target engagement, permeation, and efflux avoidance all in one shot, essentially. And so we want to predict molecules that can do all of these things. That is, they can permeate the bacterial membrane or membranes in the case of gram-negative bacteria and cell wall. They can avoid efflux pumps, and they can engage with a relevant target that will, when inhibited or interacted with, will kill the bacterium. And so our hypothesis is that we can build a single model that can learn chemical features associated with all three of these properties. We want to, though, go beyond just finding molecules that kill bacteria. We want to find molecules that are going to be progressible to drugs and to new antibiotics. And so not only do we need to kill bacteria, we need to not kill mammalian cells. We need to find molecules that have favorable medicinal chemistry features, that is, they're chemically tractable, don't have known toxicophores and other problems. And finally, we want molecules that are chemically distinct from known antibacterial compounds. We don't want to find another Me-Too, another beta-lactam or another fluoroquinolone. We want to find new molecules with new mechanisms of action. And so the implications there are we need models for E. coli activity and for mammalian cell cytotoxicity that extrapolate to new compounds. We need to filter out unfavorable medicinal chemistry and Payne's molecules, these pan-assay interfering compounds. And we want to remove molecules structurally similar to commercialized antibiotics. As John said, there's lots of molecules out there. There's a paper that estimates 10 to the 33, but as John says, there's multiple estimates of how many there are. In any event, it's a very large number, way more than anybody can do anything with. Commercial screening collections that you can buy, from Molport or MQL or other compounds that'll send you a plate of compounds to screen are around 5 million compounds in size. And importantly, we want to be able to cherry pick out the most active compounds in these collections. We don't necessarily need to predict an MIC. We just want to say, of these 5 million compounds, here's, the top 50,000. And so in order to predict well on these, we want to maximize the amount of data we can use to train these models, because we want access to the largest and most diverse set of activity data we can use to train a model on. And this is where we've done things a little bit differently. Well, first, let me tell you about how we design and test these models, and then I'll tell you about sort of how we've done these a little bit differently. So we want to prioritize compounds for screening. That means we want to be able to predict compounds that are structurally dissimilar from those that we train on, right? We don't need, if we have a molecule in our training data, we don't need to screen molecules like that because we know something about them already. We want to make predictions about the molecules that don't look anything like the ones in our training data. So we scaffold split our data sets for evaluation. So we hold out our evaluation or test data from chemically distinct molecules as defined by the Bemis-Merco scaffold, which is an old cheminformatics approach. And then we evaluate on that test set and evaluate the model by how well it prioritizes active molecules. And so we generate these virtual screening plots shown on the right, where we look at what fraction of the hits we find in that test set as a function of how much of the library we screen. And so you can evaluate something we call lift at X, which would say, if you find 20% of the hits after screening 1% of the library, that's a 20x lift compared to what you would do in the absence of this model. You can also evaluate by the Gini coefficient, which is just a restatement of this that says, how efficiently do you find these compounds. A perfect model would have a Gini coefficient of 100 and random is 0. All right. So one thing that we noticed early on in our work here is that antibiotic activity can be predicted across strains. And this is a somewhat complex slide, but the bottom left here is a large data set from COAD where they've conveniently evaluated the same set of molecules in a wild-type E. coli, a Tol-C efflux deficient strain, and an LPXC permeabilized mutant. And what you can see is that the vast majority of molecules that are active in that wild-type bacterium are also active in these deficient strains. The converse is not true for the reasons we've discussed, but basically what we realized is that in any screening collection, something like 98% of compounds are inactive in any strain. And So we can use that data from these efflux deficient strains to say something about wild type activity, even though they're not perfect predictors, because the vast majority of vast majority of compounds are inactive, and they're inactive in all strains. And so what we actually used John Stokes' first paper with his training data, and thanks for making that public, as a test set here, because that was run in a wild type E. coli. And what we did was trained a model to predict activity based on 400,000 compounds that we had measured in a permeabilized E. coli strain. And we showed that outperformed, shown in red, the predicting this wild type data compared to a much smaller data set we had measured in wild type E. coli. And this I was initially surprised about. We've thought about it a lot now, and it makes sense to me. But at the time, I thought, gee, this is really surprising that you can get this cross-strain predictivity. I think we now understand why that is. But that gave us sort of a foothold into maximizing the amount of compounds we could train on, because what we then realized was that you could take this a step further and not only train on one data set, but we could leverage multiple data sets measured under multiple different strains and multiple different assay conditions. And so shown here in these bubbles are various publicly available E. coli data sets, plus in the dark blue on the upper right, our internal screen that we did before we started training these ML models. We did a 400,000 compound HTS screen. So across all these different assays, there's data on a little bit over 1 million compounds for E. coli activity. Some of it's MIC, some of it's percentage inhibition and single point screening. And we wanted to use it all. And so you can't put it together on the same scale because it's measured in different strains under different conditions. And so what we did was to pick a technique from machine learning where you train a model that doesn't predict a property, but it predicts a ranking. And so what we realized is because we care about picking out active molecules, we care about being able to preserve the relative ordering within these data sets. And so what we do basically is assign a rank to every compound in each one of these data sets. We can treat each data set independently. We don't need to be able to compare across data sets, only within data sets. And so we train a model to rank across data sets, one model that can correctly rank compounds across all the data sets, or can correctly rank all compounds within each data set for all data sets. And so this is called a multitask ranking model. So within each of these 9 data sets, it's learned to properly rank compounds within that data set. And what we can show, shown here, is that in all cases, this model performs as well as a regression model or a classification model trained on a comparable data set or a scaffold split version of that data set. So we'll hold out 20% for testing on train on the other 80%. And what we find is that our ranking model performs as well on every data set we've trained it on as a model trained just on that data set. So in the left-hand panel here, you can see we're comparing our LPTD classifier trained on our in-house data in blue, and you can see the performance of the ranking model trained on all 9 of these models, or all 9 of these data sets, works about as well. Conversely, a classifier trained on a wild type data set shown in yellow significantly underperforms that ranker, although it outperforms random. The converse is true on a wild type data set. So again, the ranker performs similarly to a classifier trained on wild type data, whereas now the LPTD classifier underperforms because it's trained on a different strain. And so this was quite striking, but very, very nice for us because it means these ranking models can be used regardless of what strain we want to predict on. So the ranking model performs equivalently to the single test models on the data they're trained on and better on other data sets. So it's always as good as a single test model and often better. All right. So these we can also show enrich for hits in early selection. This is what we really care about because we don't want to screen, 5 million screening compounds. We want to screen 50,000. And so we care about the ability to enrich hits in that first 1% screen. These perform well there. And we can also do the same thing for cytotoxicity. And so we built, again, a multitask ranking model. This was trained across 16 data sets. These are mostly PubChem data sets that are people's counter screens for mammalian cell toxicity. So we scooped up 16 data sets, including measurements on HepG2, HEC293, and 3 other cell lines, and trained a model on about 2 million compounds across those data sets. And we see the same thing. Again, the ranking model performs equivalently to the single task models on the data they're trained on and better on other data sets. So with both these models in hand, we then made predictions of the enamine screening collections. And so here the heat map is shown in this blue-green false color. And you can see, not surprisingly, that cytotoxicity and E. coli activity are correlated, right? There's lots of molecules that are just broadly toxic. And so E. coli activity goes up as you go to the right in this plot, and cytotoxicity goes up as you go down. So the upper right quadrant is where we want to be. Those are molecules that are relatively toxic to E. coli and relatively harmless to mammalian cells. And so what we did was to pick 5,000 compounds. We cherry-picked 5,000 compounds from this diversity, or from this collection, starting from the upper right and moving in. Those are shown in the little blue circles. We limit how many structurally similar compounds are selected to maximize diversity. And we also remove any compound that's similar to any molecule that's ever been mated into phase 1 or beyond in the clinic. So we cherry-picked 5,000 of these compounds from enamine, had them ship them pre-plated for screening. We also selected a number of pre-plated sets with high fractions of predicted actives, and then had all these screened in our LPTD hyperpermeable strain of E. coli. And the results are shown here on the left. So you can see the pre-plated libraries are in the dark blue circles. You can see the library hit rate. This is the confirmed hit rate after hit validation in triplet retesting. And you can see there's good correlation between the predicted library score based on our models and the number of active molecules against E. coli. And you can see our cherry-picked model, or our cherry-picked compounds, have the highest hit rate, and it's about a doubling of the hit rate over our unguided screening we did for this 400,000 compound library early on. And so we got twice as many hits while selecting for low cytotoxicity and maximizing diversity, and we got significantly more highly active compounds and fewer weak inhibitors compared to our other screens. We then went on to follow up with cytotoxicity counter screening for the actives in this collection, and we find that we get about a threefold lower failure rate due to cytotoxicity. So only 13% of our ML selected hits failed due to poor cytotoxicity compared to 37% of our unguided selection. And so we see about a 3x gain in progressible hits from our machine learning guided screening. And even though we didn't select for this, we also identify a larger fraction of wild type active compounds in our ML guided screening compared to our unguided selection. So we were very pleased with the results of the screen. It outperformed, it significantly improved the number of molecules we got out that were interesting to move forward with. Because remember, we also removed everything from this collection that was similar to a known antibiotic. So all of these things were structurally dissimilar from anything that's made it into the clinic before. We then went back and did a second round of screening, cherry picking 25,000 molecules and saw similar results. And then here's like a map of chemical space, looking at all screen compounds across all of our screens, showing our ML guided hits in blue, approved antibiotics in green, and then non-clinical antibacterials with known mechanisms in red. And you can see our blue hits do indeed fall in different parts of chemical space. So they're chemically and structurally distinct from molecules that have known activities. And it gave us lots of starting points to look at for moving forward. So those of you who are familiar with the space will notice that these hit rates are lower than have been published for some of these other models. And that's because we were applying all these different filters in here, removing things structurally similar to known antibiotics, counter-selecting for low cytotoxicity. And we estimate if we just look at the top predictions from our E. coli activity model alone, we would have gotten a much higher hit rate, of something like a 30% hit rate, which is comparable to what's been reported in literature by Stokes, by John, and others. And basically what's cutting our hit rate down to these lower levels are all the counter filters we're putting in place so that these molecules don't drop out as we want to move forward through the drug development process. So we're pretty happy with that performance. So finally, then we go into our imaging platform, and I'm not going to talk about this in detail, but this is derived from work of our founder, Zemer Gattai. And so this is an image-based phenotyping platform where we grow E. coli, treat them with antibiotics of known mechanism, about 150 different molecules covering a few dozen mechanisms, about 30 mechanisms, and then stain an image in high throughput with 100x oil immersion imaging. And you can see on the right here, this is a UMAP projection, so just a qualitative projection of the high dimensional phenotypic space we map out into a 2D plot. We do our actual classification using supervised machine learning, but this is a nice way to look at the data. And you can see that the phenotypes we observe in these images break down nicely along mechanism. And so we can separate out compounds by mechanism and predict compounds that have known mechanisms, and then make predictions about compounds that fall outside of those known mechanisms or outside of those known phenotypes as having novel mechanisms. And so we ran all the hits that came through our screen through this platform and identified a large number of molecules, a few dozen that were predicted to be phenotypically novel. And then we went and did resistant mutant generation to identify targets for many of those. I'm not going to talk about any of those results in detail, but just here's a few examples. So we've done now these 2 rounds of screening of 104,000 compounds, 30,000 were cherry-picked, 70,000 were pre-plated libraries. We got about 3,000 primary hits. We selected 1,000 for follow-up. 300 of those were active in our in-house dose response assays. This seems to be an unfortunate side effect of using this LPTD strain, because other organisms and other strains we screened in show much higher follow-up reconfirmation rates. And here's 3 compounds that I picked. Again, for IP reasons, we're not showing structures, but you can see there's, we're getting MICs out of these screens of, single-digit microgram per mil. We did our screen in minimal media, and so we actually found a lot of targets in various biosynthetic pathways. So here's 2 different pathways in amino acid biosynthesis and then a uracil biosynthesis pathway. And all 3 of these compounds show no HepG2 cytotoxicity at 100 micromolar. So we see very good windows between the cytotox and the E. coli activity. Because of the targets, these are not super appealing for a clinical antibiotic because they wouldn't be essential at all sites of infection, but they are very nice validations of the platform. And we've got 3 molecules that we have identified that do have very attractive targets that were predicted novel by our imaging platform and subsequently validated as hitting a clinically novel target by resistant mutant generation. 2 of those we've paused for business reasons, but one of those, AP01, is in lead optimization, and we just received CARB-X funding to help advance that program to the clinic a few days ago. So we're very excited about that, and that validates, at least at some level, that we can use this to get molecules that we can advance to drug development. And I believe that's my last slide. So just to conclude here, we've shown that we can train predictive models for both antibacterial activity and cytotoxicity across data from multiple assays, including across strains and media or across cell lines and assay conditions. We've used this for a couple of other targets internally at ArrePath and have seen consistent good performance of these learning to rank approaches. Using these models for high throughput screening increases the antibacterial hit rate by twofold and reduces cytotoxicity failures by threefold. It also increases wild type hit rate, which suggests or indicates that the models learn features associated with permeability and efflux avoidance. We can prioritize for mechanistic novelty by using imaging, and we've used this to advance molecules to late stages of drug development, and aim to bring a molecule to the clinic in the next year or so. So thanks everybody for your attention, and thanks to the staff and the team at ArrePath that's made all this work possible.
Kurt, thank you for the terrific presentation, and I'm seeing our questions section here filled with fantastic questions. So I will dive right in, and first we'll start with questions for John Stokes from the earlier presentation. So the first one for you, John, is, and John, if you're able to pop up on video, perfect. So I'm just reading these directly from the attendees. The first one is, wouldn't it be better to make the output gradual instead of binary labeling? So apply regression instead of classification, as some compounds seem to inhibit growth better than others in the training data. Great question.
Yeah, it's a perfect question. And we talk about this all the time in the lab. So we've tried to train regressors, and we still try to train regressors using the training data that we acquire here in the laboratory. So this is like a practical problem, right? So again, I'm going to go back to what I said earlier. I'm an early career researcher and broke. So if I had more money, right, to gather larger training data sets, we would probably train a regressor, right? Because that incorporates some level of potency, right, into the prediction. But given the size of the training data that we often work in, we're talking like, 10, 20,000 compounds. Our regression models significantly underperform binary classification models. So we treat this practically as a binary classification task for most of our antibiotic discovery applications. Not all. We have a couple where regressors look to be pretty good, but for most of the time we do this as a binary classification task. And that's fine, right? So because I come at this from, the perspective of these ML models are just like suggestion generation boxes, right? We ultimately have to take those suggestions, bring them into the lab, and see what works and what doesn't. So if a binary classification model for some bug and some set of properties gives us like a reasonable number of like good shots on goal, such that when we bring them into the lab, we have like a reasonable number of molecules that show in vitro success, like that's good enough, right? So that's the long answer. The short answer is, could I train a regressor? Yeah, we could, but we need more training data most of the time. And binary classification models seem to be working good enough for now. When they start failing, we'll switch to regression tasks and figure out the fancy footwork on how to do like better, I'll say like data extraction or data representation from, our smaller training data sets.
John, another question for you. So an antibiotic must have three characteristics: antibacterial activity plus safety plus pharmacokinetics. You described training for antibacterial activity, but how do you train for pharmacokinetics and safety? And Kurt, I know you touched on this a bit with cytotox, but if you have some thoughts on that as well.
Yeah, so that's like the perfect question. And when I teach this type of stuff to third-year undergrads. And I always teach them finding an antibacterial molecule is easy. Finding an antibiotic is super hard, right? Because things that are good antibacterial molecules are like fire and bleach and stuff, right? But I can't turn fire or bleach into an antibiotic. So this goes back to the idea of antibiotic discovery inherently being a multi-property optimization task. You need a single molecule, right? Most of the time, a single compound with a structure that satisfies 1,000 different properties required such that you can give it to a patient. And what that molecule will do will kill the infection before you kill the patient, right? That's fundamentally the problem. So something I didn't talk about, it's like we're now incorporating these million cytotoxicity data sets into our modeling. We haven't done any like PK, PD type stuff because I haven't found data sets that, in my opinion, are robust enough to model these properties appropriately. But yeah, we do regularly employ like in-house gathered cytotoxicity data to help us prioritize molecules. I just didn't talk about it because I was going to run out of time. But it's a perfect question. And for all the folks here listening, like antibiotic discovery or design, however you want to think about this, you're trying to take a single molecule and optimize it good enough across a ton of different properties to satisfy the use case that I can give it to a patient and the patient will be fine, but I'm going to kill the infection, right? And I'm sure like I don't know, Kurt's smarter than I am. I'm sure you can build on that answer for sure.
Yeah, I think it's exactly right, and I would just add, it's unrealistic, in my opinion, to think that you're going to be able to run these kind of models and get the antibiotic out from that initial screen, right? Chemistry and medicinal chemistry is just way too complicated, and we understand it way too poorly to do that, right? Maybe if we had data on hundreds of millions of molecules for PK, PD, toxicity, all the things you care about, you could do it, but we don't. And so, kind of the best you're going to do is find a good starting point, right? Find something that has a good mechanism, isn't overtly toxic, and then you can optimize it for the other parameters you care about, right? But I think people who are new to the space of ML think like, oh, this is going to be a magic box. We'll get molecules out of it, and it's just going to give us drugs. And if we knew way more about what made a good drug, you probably could do that. But we are maybe never going to know enough about what makes a good drug to do that, or not in my lifetime anyway. So so it doesn't eliminate the job of medicinal chemists and drug discovery. It just helps you get to a better starting point.
Yeah, I couldn't agree more. And, I just want to add one piece because it's so relevant to our work right now. So we're currently incorporating this, multi-property optimization that John mentioned and open accessing data around 8 or so parameters that are meant to get to better drug-like candidates coming out of our AI platforms. And so our hope is that researchers worldwide can use this data to, incorporate extra or additional down filtering into their own models. So that it's not just the purview of big pharma that has these massive data sets, but that we can all kind of lift all boats in this space. So TBD on exactly when we'll release that, but we'll make sure to get it out to all researchers who are interested. So in terms of additional questions here, John, a quick one for you. So when will the structure of enterolalan be made public? I know that's very hard to say. All of us have negotiated with, lawyers and IP groups and universities, but in case you have a timeline on that.
Yeah. So great question. So I'll say this, the paper is under revision. I expect, unless like something catastrophic happens and unexpected, like the revisions will be done by like June. And then, if editors and reviewers don't hate us, like the paper will come out a little bit after that. So I don't know, maybe like end of the summer, I hope. We'll be able to, you'll see it in the publication and I'll let everybody know. I don't know, like I'm not a medicinal chemist, but when I look at the structure of like this molecule and the analogs that we've started to now design, it's like, I always have like an internal, like, would I eat this sort of dialogue? So like you look at the structure of a molecule, it's like, would I eat this? And like my answer tentatively now is like, probably, you know what I mean? So it's like, it's not crazy. It's a cool looking structure. So yeah, end of the summer, I hope. Unless like catastrophe ensues, in which case I don't know.
A question for you, Kurt. So what is the basis for saying that a multitask ranking system is better than the single task system?
I guess I think how I would frame it is that it's not better than the single task ranking system on a specific task, but it's more general purpose. So on any single task, what we tend to find is that the ranking model performs equivalently to a single task model trained on that task, but it also works as well as single task models trained on these other tasks. And so it lets us have one model that understands this diversity of chemical space and presumably generalizes better than having, in our case, it would have been 9 different, single task models for bacterial activity. We do have other cases. Yeah, so in general, I think that's the claim I would make. We have other cases where we can show that ranking models do perform better on our in-house data compared to single task models trained on a single data set. And that's probably due to a generalization ability.
And Kurt, another one for you. Can the learned features from your models be rationalized into permeation and efflux avoidance properties within a defined set of physiochemical properties?
Yeah, we've tried doing that. What we find are basically, the answer is to some extent yes, and we find the same things that other people have found, right? So we find, features like log p, other features that correspond to hydrophobicity and things like that, stuff that's similar to what's been published, for example, by Paul Hergenrother and his entry rule. But we generally find that there's more features beyond that that are hard to rationalize. And we have more or less stopped trying and just treat the models as black boxes right now.
Question for you, John. Are you planning to revisit the entry rules in gram-negative bacteria based on ML? And this attendee says, cool stuff you're doing.
Yeah, dude, perfect question. So I have this an undergrad in my lab, dude named Wesley, who's just like a total killer. And I'm super lucky. He's staying in my lab to do a master's degree, and hopefully I can convince him to transfer. But yeah, so he's working on a project to train models that can optimize molecules for outer membrane permeability that are they're not going to be interpretable. Like, I fully agree with Kurt. Like, understanding the molecular features that dictate whether a small molecule can get through an outer membrane or not is like super challenging. And kudos to the Hergenrother group for the entry rule stuff and the pseudo work and everything, like super rad. I just think that if we treat this largely as like a black box, right, and we allow the model to do optimization around every individual input scaffold based on what will optimize that specific scaffold, I think the probability of success is going to go up. So yeah, I encourage you to if you go on my website and go to the people page, and there's a guy named Wesley Ta, just send him an email. Like, he'll geek out about this stuff forever. So I highly encourage it. So yeah, Wesley's working on it, and he's super smart, like way smarter than me. So I'm sure he'll figure something out.
Yeah, and
Well, these questions. Sorry, Kurt.
I was just going to add to that approach tends to be how we've worked at ArrePath. We've used our E. coli ranking model. We've also trained some permeability or accumulation models based on public data, and we use those to optimize early stage molecules. And then once we get enough data, we start training models that are specific to the chemical family we're trying to optimize. And we're right now working on making hybrid models that try to learn across all the data, but focus in on predicting a specific chemical series.
And we have another great question from the audience. I'm very curious your take on this, Kurt. So, Kurt, what is the cost per hit comparing machine learning / over cherry picking versus, sorry, let me say that again. What is the cost per hit comparing machine learning / cherry picking versus your previous diversity library screening?
Yeah, that's a great question. One of the things that really surprised me when we did this is cherry picking can actually be quite cheap. So part of the reason we screened so many pre-plated libraries is when we first set out to do this work, we thought it was going to be cost prohibitive to cherry pick individual compounds. But it turns out if you're cherry picking a few thousand molecules, it's only a couple of dollars per compound to cherry pick into pre-plated assay-ready plates. So we've not run the numbers, but it's not a huge cost, and we outsource our high throughput screening, and that is at least as large a cost as the compound procurement. So I don't think it's a huge difference in price per hit. It might be cheaper. It probably is cheaper for the ML screening, but I think it's not, for us, not so much a cost savings as a quality improvement, that we have fewer molecules that fail to progress when we run the ML guided screens as when we do an unguided screen. In our unguided library, we found tons of stuff similar to known antibiotics, tons of stuff that failed for cytotoxicity because it was just membrane disruptors or other generically toxic molecules. And so those problems get a lot better when you do the MO guided.
And Kurt, we couldn't agree more at Fair Bio. I think our feeling is not to try to reduce costs at these early stages, but really reduce the cost of failure. Take fewer shots on goal, but better shots on goal, and that will significantly reduce that 1.3 billion or so number that we hear oftentimes for getting a drug across the finish line. John, a question for you, a little bit of a longer one. So this attendee says, very cool work. Are not most natural product sources for potential novel antibiotic classes not well characterized in training data that might be used for such AI modeling? Could you speak to the need and value or not of continued bench research to enrich the training data? And then a little bit of a follow on here. Is the Synthemal model equally able to predict the drug ability, e.g. Lipinski rule of 5 and the potential toxicity of the molecule? Or again, how much then requires investing in bench research to complement the AI modeling work?
Yeah. So I'm a microbiologist by training, right? Like I spend the first, 10 years more of my professional academic life, like pipetting, right? Like I'm a total noob when it comes to this MLE type stuff. So 100%, like drug discovery is such like a human endeavor. Like you're never taking people out of this, this process, right? ML is simply like another tool in a drug discoverer's toolbox to help them make better decisions as they're, advancing some compound towards something that you could give to a patient to treat whatever disease you're most interested in, right? So let me answer the Synthemal one first. So the short answer is yes. Synthemal can like relatively easily be adapted to incorporate like an arbitrary number of molecular properties, and the algorithm will design molecules that satisfy, as many of those properties as possible. The guy in my lab who co-first authored the original Synthemal paper is a guy named Gary. You can reach out to him. We actually have Synthemal version 2, which is way better, that we're finishing up the paper for hopefully in the next 2 weeks. And we're going to preprint that one. So if you just want to send me an email and say, hey, I asked about Synthemal, I'm happy to chat and I'll send you the preprint when we're done it. And what was the first question? That was a good question too, but my brain isn't working.
Let's see here. So are not most natural product sources for potential novel antibiotic classes not well characterized?
Yeah, 100%. And natural products, I'm sure as you know, are the lion's share of our currently clinical used antibiotics, largely thanks to the Waxman platform, right? So, when we gather training data sets, we incorporate all of those known natural product antibiotics in our training data, right? Unfortunately, large natural product molecules are not readily synthesizable or often easily purchasable, right? So it's a bit of a blind spot, at least the way that we currently work, in terms of acquiring large natural product type molecules to bring into the laboratory. Sometimes by accident, when we're running predictions on these very, very large chemical spaces, there are subsets of those chemical spaces that are natural product which we can acquire for relatively little money. But I do think, there's a huge need for folks to develop, bench lab methods. And whether these are synthetic biology approaches or really sophisticated med chem semi-synthesis approaches, to give, modeler types, easier access into synthetically tractable natural product chemical spaces. Because so far as I know, maybe Kurt's seen something else, I haven't seen anything quite like that. And that's so far outside my wheelhouse that I wouldn't even know where to start. So we're relying on folks, who have remarkable synthetic chemistry and synthetic biology chops to help us get into these novel natural product chemical spaces where we know antibiotics can live. Does that make sense? I hope that made sense.
Absolutely. And I think, John, this is probably for you, given that you mentioned, disinfectants and bleach and whatnot. How about actually diversifying and also exploring disinfectants that work on biofilm?
Was that a question for me?
I think it's for you, just given
Sure.
that you mentioned some of the disinfectant,
Yeah.
concerns with mechanisms.
I'll say, like, if you can gather training data to train a model to do a thing, the model will do the thing, right? So if you're interested in identifying like structurally and potentially functionally novel molecules that are like really, really good disinfectants specifically against like surface embedded biofilms, you can do it. It's just another, like property prediction or molecule design task. The limitation is not in the modeling. I don't think. I think the limitation is in the ability to acquire sufficient quantities of high quality and biologically relevant training data, right? So if you're a bench scientist that has that capability and the necessary expertise in, biofilm physiology, like go gather the training data, and then, if you don't want to model it, we'll model it. You know what I mean?
Absolutely. And if folks have more questions in the chat, of course, submit those. And I have a couple questions for you both. Given that we're all working in this rapidly, evolving field, it's, every day there seems to be a new innovation in Gen AI and AI for drug discovery. I'm curious what you're experiencing in terms of bottlenecks, where you know that if you had this, whether it's resources or access to certain models or certain data sets, you could unlock certain efficiencies and really move towards your goals faster.
I mean, I think the big thing is data, right? Like ML needs lots and lots of data, and compared to the places where it's been super successful, drug discovery is pretty data poor, and the data is mostly locked up in big pharma, right? If you look at, probably the biggest success in AI for biology is things like AlphaFold, and that's enabled by the fact that the, protein structure community organized itself 50 years ago and put all their structures in a database, right? And if we'd been putting everything else in a database for 50 years, we'd be in a lot better shape for training models.
Yeah,
Absolutely.
I agree. Like biological data is like inherently like difficult and expensive to acquire, especially when you're trying to do it really, really precisely, right? And the other thing too with biological data is, a screen, like let's say a chemical screen against a bug, some E. coli, like you might have two completely distinct data sets, like, but they're both E. coli antibiotic screens. But the chemical spaces that are explored and the experimental methodology through which that data was acquired, those data sets like might not be combinable to train a model across both, right? So we have to be super careful if we want to do this properly about like when we're leveraging publicly available data, for example, like are we making the best decisions about which data sets we can combine and which we have to kind of keep separate? But I 100% agree with Kurt, right? It's a biological and biochemical data limitation issue. But what's kind of cool, and I don't know, this might like, I don't know, people have different philosophies. Like, I think it's kind of rad to be like work in a space where like you're inherently sort of like data and resource limited, because it forces you to think about these problems like a little bit like more deeply and come up with creative methods about like how do I like maximize like data extraction from an inherently small data set in order to get a model to do a thing that I actually want it to do, right? Because like in some cases, like you can't just like throw money and resources at these problems. You know what I mean?
Yeah, and John, that brings up another question I have for you both. And as the human intelligence piece seems to really get at what you're talking about, John, like really finding and working with folks who are exceptional curators and validators and analyzers of data. And I'm curious, over the last few years that you both have been innovating in this field, how the teams that you hire have changed. I know in the early days of AI for antibiotics, it was often, biologists and chemists who then learned the machine learning tools. And I'm curious if that's changed in any way, and if really, the key now is true data scientists who come at this from a, more kind of data analytics approach.
Yeah, at ArrePath, we decided when we were hiring to focus more on people with ML experience. So we were quite lucky. We hired a lead data scientist from Phillips 66, actually, who'd been doing ML on petroleum compounds. And he's sort of our main innovator in the space and has brought a lot of these techniques to us. But then the other folks we hired have largely been from non-chemistry, non-bio backgrounds, because we felt it was more important to have people with good ML skills and teach them the chemistry they need to know than the other way around. And we feel like that's paid off pretty well. So, I have one guy on my team from Capital One and a former astrophysicist and another person. So that's worked well for us. But we have the luxury of having, very senior medicinal chemists and very knowledgeable people elsewhere at the company to help give us the context we need.
Yeah, I don't know.
John, I'm
Like,
curious your take on that because I know you've had strong feelings that it's harder to teach the biology, and I'm curious if that's evolved at all over the years.
yeah, I mean, I don't know. I haven't been hiring people for very long. I still don't know what I'm doing, but I don't hire expertise. I hire people, right? Like all I care about is like, if you're interested in doing this type of work, that you're like fully dedicated and understand like the social and scientific importance of it. And then I'll let you find like what you're interested in. If you naturally want to gravitate to the ML, like if you're dedicated enough, you're going to learn it anyway. And if you're super more interested in like biochemistry and microbiology and like mouse experiments and this type of stuff, you'll gravitate to that and you'll learn it anyway. What's most important to me is that like everybody on the team has to be multilingual, right? You have to be able to talk to a computer scientist and you have to be able to talk to a biologist and you have to be fluent in both. But you're going to have your own expertise like across these domains. Like you might be a super like genius microbiologist and sort of just like dabble in ML or vice versa. So I think the key across drug discovery, but this is true for antibiotics too, it's like you need people on teams that have like very diverse expertise, right? Because they have to understand how their specific like ultra expertise fits into the whole process, right? So yeah, you just need people like I hire people who are obsessed with AMR, right? And then I let them find sort of like their specific interest, and then they figure out how to like do it, like people are smart. Like this isn't rocket science, right? Like they'll figure it out.
And I have a question for you both on, kind of where the future is heading. So, what are you most excited about in terms of what your teams are building and milestones that the audience may look forward to coming out of your company, Kurt, and your lab, John?
I don't know. We're doing a ton of like just rad stuff. I don't think there's like any one thing that I'm most excited about. Like I'm not an ML guy. Like I'll just say it. I'm interested in solving the AMR problem, right? So it's like, we're throwing all of our expertise, and right now it happens to be like heavily embedded in ML to try to, as efficiently and inexpensively solve this challenge, right? But that doesn't mean that ML is the be all and end all to fix this problem. It's like I said, it's another tool in our toolbox to help us move this in the right direction, right, towards solving this problem. Ultimately what I'm most excited about is having like a super robust pipeline, right, both preclinical and clinical, of structurally and functionally novel drugs that can like cure patients who otherwise would have died. That's what I'm interested in. And it's like, however, us as like a global community of engaged scientists like want to get us in that direction, it's like we all need to be doing something, right?
Mm-hmm, mm-hmm. No, we couldn't agree more. Very much share your vision, John. And Kurt, any thoughts on that?
Yeah, I think, we share the same goal, right? We ultimately want to bring molecules to the clinic to treat patients, right? That's the whole point of doing this. And so, we're excited to see progress on our lead program. We really hope, that we'll get into the clinic in a year or two. And then I think the other hope is that, these ML tools, right, as I said, they're not a panacea. They're not going to solve all the problems we have, but they should make drug discovery more efficient. And we all know that drug discovery has gotten more and more expensive. It's gotten harder and harder to get new drugs. And hopefully this helps bring that cost down somewhat, or at least stops it from continuing to increase.
And Kurt, a follow on to that, and I know those of us on the industry side get this virtually in every panel. How do you contend with these challenging headwinds in AMR from a market perspective?
Yeah.
We're very much any group that succeeds is a success for the whole field, we feel. But we really are waiting for those signals that there is a marketplace that's profitable. so that more early stage companies can really enter this space?
Yeah, I mean, I think it's going to be hard absent large scale changes in how we pay for antibiotics. So we're doing a couple of things. We think that our lead program has the opportunity to be a hospital avoidance strategy, and so we think that potentially can tap into some markets that are underserved. And then we're very rapidly moving towards compounds to treat non-tuberculous mycobacteria infections. That's another area we think is underserved and where we think the economic incentives for those molecules are better aligned with the clinical value. But we're also looking to branch out into areas beyond antibiotics, solely for commercial reasons, because that's where the money is. So, at some level as private industry, we're going to follow what the economic incentives are. And so, we need to change the way we pay for antibiotics in order to make that, commercially appealing. But of course, also non-dilutive funding is tremendously helpful here, right? Like we wouldn't be able to do our lead program without the money we've gotten from funders like Carbex and PACE. And so, we would be very happy to have a, clinically or a commercially lucrative, side doing, non-antibiotic development and then a non-dilutive funded, cost neutral antibiotic arm. I wish we had more, we've got really interesting molecules, but there's just not the money to develop them. So I wish we had more funds to do that.
And Kurt, another question for you for one of the attendees. Congrats on Carbex. The release states that you have both IV and PO. What is the PK?
We're still working on that, but we have evidence that we have, tractable oral bioavailability in the series. So we don't yet, we've not yet got the molecule. Otherwise, we'd be, have nominated a development candidate. We'd be telling you that. But, we've got molecules with good oral bioavailability. We've got other molecules with very potent MICs and, good PK. We don't have, the molecule that combines all those features into one molecule yet, but we feel like based on all the data we've seen, it should be very, very doable.
That's great. And then last question for you both in terms of the clinical pipeline. So John, you mentioned this, that, Acinetobacter is one of your favorite bugs to address for clearly the unmet need, and then also the scientific progress you're making in your lab. I'm curious if you have, and I know these are hard questions to prioritize amongst all of which are urgent unmet needs, but if you think there's one major area of the global pipeline that needs replenishment, I would say in our case, we really think Pseudomonas, and anything that is able to permeate the cell membrane for Pseudomonas and be effective. I'm curious if there are any other calls to action that you both have, especially for all the folks on this call, in terms of new programs that are urgently needed.
Yeah, I mean, I'm going to say what everybody already knows this: broad spectrum gram negative, right? Orally bioavailable if you want to be super fancy, but I don't think that's like a necessary. So yeah, if you have something that can like kill gram negatives with a good MIC and has all the other properties that you need to make a drug, I think you're sitting in a pretty good spot, right?
Yep. And that's what we feel like is, broad spectrum gram negative is the place to go now. I think, there's a lot of interest, especially academically, on these narrow spectrum antibiotics, but they're hard to use in the clinic because you don't have the infrastructure for testing for source of infection. And so, like a lot of places, there's a disconnect between what you'd like to do in the ideal case and what's sort of feasible in the world we inhabit right now.
Well, I think we are just out of time, and I want to say a huge thank you to you, John and Kurt. It was just a brilliant discussion, far-reaching, and I think technically one of the deepest dives in AI for antibiotics that I've been a part of. And as always, great seeing you, John. Wonderful to meet you, Kurt, and thank you to Revive and GARDP for having us all. And hope we'll have more follow-on discussions on this rapidly moving and incredibly exciting space.
Thank you all.
Yeah, thanks everybody.
Great.
Thanks everyone.
Thank you very much. Thank you very much, Akhila, for moderating today. And thanks a lot, Kurt and John, for your great presentations. So we'll be announcing our next Revive webinars very soon. Please keep an eye on the Revive website and also subscribe to our newsletter to find out when this is released. And also follow GARDP on our social media channels sit here to keep up to date on our future announcements. So that will be all from me and from all of us. Thanks everyone for joining today and for contributing to the discussion. I hope you found the webinar interesting and useful, and that you'll join us again in future. Please spread the word within your networks and join us next time. Thanks again.